For a flowering plant to reproduce, it has to manufacture and successfully deploy a microscopic, chemically armored genetic payload. That manufacturing process happens here, in the andrecium, the male reproductive organ, which is made up of individual stamens. If you look closely at a single stamen, you'll see two distinct physical structures. At the base is the filament, a long, slender stalk. Sitting at the very top is the terminal head, called the anther. The filament serves a purely structural role, elevating the payload to the optimal height for dispersal. The anther is where the actual biological work happens. It operates as a highly specialized bioreactor, engineered to convert raw diploid plant tissue into millions of independent male gametophytes. To understand the engineering of this bioreactor, we take a transverse section, a horizontal slice straight through the anther. Looking at this macrogeometry, you can see it is naturally divided into two distinct halves. Because of this, we call the anther bilobed. If we zoom in on just one of those lobes, we find it is further divided into two separate chambers, making the overall structure dithecous. Two lobes, with two chambers each, gives the anther its characteristic four-sided, or tetragonal shape. Positioned directly at those four corners are the microsporangia. These are the core incubation chambers for the developing pollen. A central band of connective tissue runs through the middle of the anther, holding the entire structure securely together. Surrounding each microsporangium is a highly specific wall structure. The outer boundary consists of three layers: the epidermis on the very outside, the endothecium beneath it, and the middle layers. These three outer layers are mechanical. They provide physical protection for the developing cells inside, and eventually they facilitate the rupturing of the anther to release the payload. But the fourth, innermost layer, the tapetum, is distinctly different. The tapetum offers no physical defense. Instead, its cells are dense with cytoplasm and often multinucleated. Its sole function is to secrete nutrition directly into the developing microspores. Every layer serves a distinct architectural purpose. The outer walls are built to defend and break, while the inner wall is built entirely to fuel the core. Inside that tapetum boundary lies a dense mass of cells called the sporogenous tissue. At this early stage of development, the sporogenous tissue is completely diploid. It carries the standard full set of plant chromosomes. As the anther absorbs nutrients from the tapetum, these cells grow and mature into microspore mother cells, also known as pollen mother cells. To create viable reproductive units, these diploid mother cells must undergo meiosis. Through meiosis, a single mother cell divides twice, resulting in a tightly bound cluster of 4 cells known as a microspore tetrad. Because of this meiotic division, each of these 4 microspores is now haploid, carrying exactly half the genetic material of the parent plant. As the anther reaches full maturity, the internal environment changes. The tissue begins to lose water and dehydrate. This dehydration causes the microspore tetrad to dissociate. The 4 cells separate from each other, floating freely within the sporangium. Microsporogenesis solves the core mathematical problem of plant reproduction. It neatly halves the genetic material to prepare for fertilization, while simultaneously multiplying the total payload quantity by 4. Those independent microspores rapidly develop into mature pollen grains. Depending on the species, they take on wildly different physical shapes, sizes, and textures. But internally, almost all pollen grains rely on a specialized 2-layered architecture. The outer layer is the exine. It is thick, highly textured, and conspicuously discontinuous. The exine is constructed from sporopollenin, one of the most resistant organic materials known to biology. It withstands strong acids, potent alkalis, high temperatures, and cannot be degraded by any known enzyme. This near-absolute chemical resistance is the reason pollen grains remain perfectly preserved in the fossil record for millions of years. But the exine armor requires gaps. These intentional openings in the sporopollenin are called germ pores, and they are essential. Without them, the future pollen tube would be permanently trapped inside and unable to emerge. Beneath the exine sits the intine. This inner layer is a continuous, flexible membrane made of standard cellulose and pectin. Moving inside the walls, we find the cellular payload itself. The bulk of the interior is taken up by the vegetative cell, which holds an irregular nucleus and massive reserves of food. Floating directly within the cytoplasm of that vegetative cell is a second, much smaller, spindle-shaped structure. This is the generative cell. The pollen grain operates as a heavily armored biological transport vehicle. It pairs impenetrable external shielding with a self-fueled internal delivery system. At full maturity, the internal walls separating the microsporangia break down, fusing them into larger continuous pollen sacs. Then, the dehydrated outer layers of the anther contract and tear open. This is dehiscence, the mechanical rupture that sheds the pollen payload into the environment. In 60% of angiosperms, pollen is shed carrying just the vegetative and generative cell. But in the remaining 40%, the generative cell divides mitotically before release, forming two distinct male gametes. These shed a mature, 3-celled pollen grain. Whether deployed at the 2-cell or 3-cell stage, the payload is strictly calibrated to ensure the male gametes are prepared for fertilization the moment they reach a receptive stigma. Once shed, the clock starts ticking. Pollen viability, the window of time the grain remains functional, is highly restricted, and it varies wildly depending on the species. For many cereals, like rice, the window is incredibly brief. Their pollen loses the ability to fertilize just 30 minutes after release. Wheat crops operate on this exact same strictly limited 30-minute expiration timer. Compare that to plant families like Rosaceae, Leguminosae, and Solanaceae, whose pollen can maintain viability for months under standard environmental conditions. In modern agriculture and crop breeding programs, researchers frequently bypass these natural biological limits altogether. Through cryopreservation, pollen grains can be banked in liquid nitrogen at -196 degrees Celsius, completely halting cellular decay. Because the evolutionary engineering of the sporopollenin armor is so robust, we can effectively freeze these gametophytes in time. We preserve the angiosperm's genetic payload indefinitely, deploying it exactly when and where it is needed.